Hi guys, thank you for joining me for Unit 1, Chapter 3. We're going to go over biological molecules or macromolecules. Once again, if you click on this link, you can get to the guided notes. And if you're following along in the book, we're actually in Chapter 3. So Biology 2E, Chapter 3 of the textbook. What we're going to go over today is actually how these macromolecules are made. The different types of macromolecules. So there's four main types of macromolecules, and we're going to go into detail of each of those type of macromolecules and different types within those categories. So the first thing that I want to go over is organic compounds. We mentioned this last class, but this class we're going to go into more detail. An organic compound is anything that has carbon in it. Now, carbon is a really unique molecule or a unique element because if you look on the periodic table, it's relatively lightweight, it's very small, but it can make 4 different covalent bonds, which means it is really powerful for making these long chains because it can make all these different bonds. So one carbon can make 4 different covalent bonds. If it's linked to all hydrogen, that would be an example of methane. But if it's linked in these long chains, it can do many different things. It can also be linked in a circular pattern or an aromatic ring. And these rings, when we see them in this class, usually represent sugars. So there are 4 major classes of macromolecules or biomolecules. The first is carbohydrate. You're going to find carbohydrates in plants, in breads, and sugars. Then we have lipids. Lipid is another way of saying fat. You have lipids in the food that you eat, but there's also lipids in your cells in the phospholipid bilayer, in the membrane that holds your cells together. Proteins are all over throughout your body. We'll talk into detail about how proteins are made. And then we also have nucleic acid, which is our DNA. So even our instruction manual, the DNA, the thing that gives directions to the rest of your body, it is a type of macromolecule. Now, all macromolecules have one thing in common. They have small subunits called monomers. And when these monomers link together in these long chains, we call them polymers or macromolecules. So a monomer of a protein, for example, would be one single amino acid. Once the amino acid starts binding to other amino acids, it becomes a polymer or a polypeptide, which then turns into a protein. So we're going to talk about the different monomers of these 4 different categories. So the first category is carbohydrate. We're going to find it in grains, fruits, and vegetables. The bread that you eat is a carbohydrate. Pasta is a carbohydrate. It provides energy to our body in the form of glucose. Glucose is the type of sugar or the type of carbohydrate that we use for cellular respiration. So we need to break down any of these complicated carbohydrates down into a simpler form of glucose. Now, all carbohydrates come in this kind of general formula where we have 1 carbon to 2 hydrogen to 1 oxygen, 1 to 2 to 1, carbon, hydrogen, oxygen. So you can see that they are mostly made out of carbon and hydrogen. And H2O, that looks familiar, water. So that's how plants create these sugars, is they use carbon from the atmosphere and water and sunlight. So carbohydrates are very interesting. Our monomer is going to be called a monosaccharide, a single sugar, mono meaning 1, saccharide meaning sugar. And then we have more complicated sugars called disaccharides, di meaning 2 subunits. And then we have polysaccharides, which are our long chains, poly meaning many sugars. So long chains of sugars. But we're still going to follow the same general formula of 1 carbon to 2 hydrogens to 1 oxygen. Like I mentioned, our body uses glucose, which is a single monosaccharide. It looks kind of like this if we draw it out in an aromatic ring. There are other types of monosaccharides though, not just glucose. Glucose is what we use in our body, but galactose is also a monomer or a monosaccharide, and it's found in a lot of milk. It's part of lactose. Lactose is a disaccharide, so galactose is a single part of that disaccharide. And fructose, you find fructose inside of fruit, but you also find it in table sugar. Table sugar is also a disaccharide called sucrose, and one of the subunits of sucrose is fructose. Okay, so they all have this ring shape to them, but they might have different groups attached to it. We still follow that same formula of 1 to 2 to 1. So we have C6H12O6 is our formula for our monosaccharides. Disaccharides are double sugars constructed of two different monosaccharides, and they're combined with this special bond right here. So sucrose, for example, is a ring of glucose and a ring of fructose bound together. And they're bound together in a dehydration bond. So both of them have a hydroxyl group on the end. And if we remove water from this hydroxyl group, if we remove a hydrogen from over here and a hydrogen and oxygen from over here, we end up with a single bond right here called dehydration synthesis. So this is how many sugars are linked through this covalent bond with oxygen. Hydrolysis is the opposite of that. Hydrolysis adds water to the system, and the hydroxyl from the water binds over on one end and the hydrogen binds on the other end, causing them to break apart. So we have hydrolysis adds water, dehydration takes water out. But these disaccharides are joined together with that dehydration synthesis. Here's a short video about it so you can see the moving parts. So here we have the hydrogen removed, the hydroxyl removed to form that bond together, and then we have water as a byproduct. All right, so some common disaccharides are lactose. We already went over lactose. Lactose is made up of galactose. Sucrose, table sugar. Sucrose has glucose and fructose combined. So table sugar, sucrose, is glucose and fructose. Lactose is going to be two galactoses. And then we also have maltose. Maltose is going to be your grain sugar. We use maltose to make things like beer. It's a really interesting disaccharide. But they're all formed with that glycosidic bond or that dehydration synthesis. Now, if we link a whole bunch of these together, it's called a polysaccharide. And in plants, we call that starch. And in humans or other types of animals, we call it glycogen. This is just a long chain of saccharides bound together with those glycosidic bonds or those dehydration bonds. If it's in a long straight chain, it's usually starch. If it's branched chain, it's a little bit different. It's a little bit harder to break apart. Glycogen is a branched chain. But you do see some plants make a branched chain, like amylopectin is a branched chain. So here we have amylose. Amylose is a single chain or non-branched chain starch. And then here we have amylopectin, which is a branched chain, but it's still connected with those glycosidic or dehydration bonds. Cellulose is made by plants, and humans can't really digest it. There is an organism that does digest it, termites, and some cockroaches can digest this. But that's because they have bacteria in their gut that helps them digest it. It has the glycosidic bonds, but it also has this cross linkage here. And so it makes cellulose is a very, very strong type of fiber. Resulting in this linear fibrous structure. So some myofibrils in animal cells, but mainly you're going to see cellulose in the walls of plants, in the cell walls of plants. So very, very strong type of polysaccharide or carbohydrate. Humans don't really digest this very well. Some animals can also make these polysaccharides. For instance, insects have to make their exoskeleton, and their exoskeleton is made out of something called chitin. And chitin also contains some nitrogen groups. So it's a little bit different than other carbohydrates. It has some nitrogen in it, but it's helpful for the exoskeleton of insects. You also find some chitin in mushrooms. If you want to learn more about chitin, you can Google it, or here I have the wiki linked to it. Chitin, I think, is very interesting. Some chocolate factories like to use chitin in their chocolate production to make it have a smooth finish. So chitin is the nitrogenous carbohydrate found in the exoskeleton of insects. So, how many sugar groups are present in a disaccharide? In my normal class, I would ask them to give me a number, but we just went over it. Di means two, so disaccharide. An example of a disaccharide would be sucrose or lactose. All right, let's switch gears and talk about lipids. Lipids are also long chain carbon molecules. They're made up of two main parts. The first main part is called glycerol. So glycerol is going to be the head of our fat, and it has three active sites made out of a hydroxyl here. And when we talked about dehydration synthesis, we talked about removing water from this hydroxyl group and forming a glycosidic bond. So fatty acids do the same thing. So we have the glycerol, but we also have these long chain fatty acids, which is just carbon and hydrogen. And these can be very, very long. Carbon and hydrogen, carbon and hydrogen, carbon and hydrogen on long fatty acid chains. Now, this fatty acid or this lipid only has one fatty acid chain. So we call it palmitic acid. Some of them have three acid chains connected to the glycerol, and we would call this a triglyceride because it has three fatty acid chains, but it's still bonded with this glycosidic bond through dehydration synthesis. And since it also has this double bond to an oxygen, we call it ester linkage. So it's an esterized fatty acid chain connected to this glycerol. So all fats are going to have a glycerol and a fatty acid chain, but depending on the number of the chains and how long the chains are and the properties of the chain, it will change the behavior of the fat. So for instance, here we have the formation of a triglyceride. We have our fatty acid chain. We're going to remove water in a dehydration synthesis, and then we end up with this glycosidic bond connected to the double bonded oxygen. So here we have our ester bond. This is a triglyceride because we have three fats. The saturated or fatty acids, like I mentioned earlier, are just these long chains of carbons, and they can be very, very long. And this one is a saturated fatty acid. We call it saturated because it has as many hydrogens as it possibly could, and it's formed in these very straight lines. So saturated fatty acids do not contain any double bonds. There's no double bonding here. We have only hydrogens. So it's saturated with hydrogens. And since they're in these long straight chains, they can actually be packed very tightly against each other. And anything with these saturated fatty acids usually are solid at room temperature. For instance, butter is going to be solid at room temperature. And I'll talk about Crisco in a minute because Crisco is also solid at room temperature, but it's not necessarily a saturated fatty acid. Unsaturated fatty acids are going to have at least one double bond. If it has a double bond, that means carbon is bound to carbon with a double covalent bond instead of hydrogen. So it's missing some hydrogen, so it's unsaturated. It does not have as many hydrogens. And these double bonds can cause kinking, okay? So this would be a monounsaturated fat because it only has one double bond, but anything more than that is a polyunsaturated fat. It has more than one double bond. Usually these double bonds cause kinking or like a turn in the structure of the fatty acid, so they don't stack neatly one on top of another, which means they're usually liquid at room temperature, like your oils. Okay, so here's an example of an unsaturated fatty acid that has a kink. This is a cis orientation, It has that kink, and it's going to be liquid at room temperature. Well, we also have something called trans fatty unsaturated, and the trans unsaturated, it does have a double bond here, but because of its orientation, it does not have a kink. So these are foods that are going to increase your LDL cholesterol. They're really not super great for you, these trans fats. So if you've ever heard that word trans fat, that's what it means. It's an unsaturated fat that's kind of been manipulated to be a straight chain. But normally an unsaturated fat is going to have a kink in it, which means it's not going to be solid at room temperature. Saturated fatty acids are naturally straight chained and naturally solid at room temperature. So here we have our two examples. Saturated fatty acids are usually solid at room temperature. So we have meats and butter, some cheeses. These are saturated. Unsaturated are going to have that kink in it. We have oils. Fish and avocado are also have unsaturated. Those omega fatty acids are unsaturated. They have kinks in them. So if I'm asking this question, looking at these two fats, I can tell I have the glycerol here. This is our ester right here, so we know that this is our glycerol. And here I have 3 long chains of fatty acids. I can tell that this is a triglyceride, but I can also see that there's no double bonds. So is this one saturated or unsaturated? Well, I know that this one is saturated with hydrogens because there's no double bonds and it's solid at room temperature. This is also a triglyceride because it's bonded 3 times to the glycerol, but I see some double bonds. So this is a polyunsaturated fat, and it's going to be liquid at room temperature. There are other types of fats, other types of lipids. For instance, wax. Wax is a very interesting type of lipid because it has 2 long chains of fatty acids, and these are extremely long chains of fatty acids. That's what this means. This means that there's more here that's not drawn. They're very long chains. They are saturated, so they're going to be solid at room temperature. And in fact, they're very hard. Usually these fats are very hard. Think of candle wax. But these fatty acids are not the only thing that's bound to the glycerol. It also has a phosphate group. Phosphate really isn't hydrophobic like these long chain fatty acids are. But the long chain fatty acids are hydrophobic. And if we go back and we want to talk about these saturated fatty acids, these are non-polar bonds, which means the electrons are shared evenly between the carbon and the hydrogen. So they're not going to be attracted to water because there's no electronegativity. So usually fats do not mix with water unless you like emulsify it, which is a whole different thing. So we're not going to be able to mix water with these waxes, which is what makes them so great for plants and some animals like the feather of aquatic birds, because these long chain fatty acids repel the water. They're hydrophobic. They create a hydrophobic surface where the water won't stick to it. But this phosphate group is not hydrophobic. It's actually hydrophilic. So this phosphate group gives it that unique property. So many plants and animals make these. They can also be used as fuel, like in candles that we mentioned. These can burn away and make a nice candle for you. Still a fatty acid though. Another type of lipid is a phospholipid that we would see in our membrane. So we just went over a different type of phospholipid. This is a phospholipid, but it's a wax. We see phospholipids in our body as well. Here we have a straight chain saturated fatty acid and a kinked unsaturated fatty acid, whereas the wax, they were both saturated. They were both straight chains. This is really important for the flexibility of your cells. So phospholipids are molecules with 2 fatty acid chains and a phosphate group attached to this glycerol, so this phosphate group up here. The phosphate group is hydrophilic. It loves water. But these tails, as we already mentioned, are hydrophobic. They hate water. And notice one is straight and one is kinked. meaning it's kind of solid at room temperature, but not really. It's still really flexible, whereas the wax was very solid, very solid. But if your cell membranes were made of this, your cells would be very rigid. So we have the flexibility by having one unsaturated chain and one straight chain. And these common groups are choline and serine, but it's okay. So here's a better look at the phosphate or the phospholipid. We have the phosphate at the top, and this hydrophilic head really, really loves water, which means if I have a handful of these phospholipids and I throw them into a cup of water, they're going to automatically orient to the outside. So all the hydrophilic heads, the heads that love water, the phosphate, is going to orient to the outside, and the tails are going to orient towards the inside because they're hydrophobic. They hate water. Well, if I add even more phosphates, it'll create this bubble. And this is how many organelles in your cells are made. Like a lysosome has this double bilayer, and inside is aqueous and outside is aqueous, but this inside layer becomes impermeable to water, and it protects and separates the inside from the outside. As your cell gets bigger and bigger and we add more phosphates to it, it becomes this bilayer sheet where we have an aqueous outside of the cell, aqueous inside of the cell, but we have this protective layer right here that will not allow water to cross it. And that is what our phospholipid bilayer is made of in our cells. It only works because we have the phosphate heads and the fatty acid tails repelling water. Now, I already mentioned that they have one straight chain and one unsaturated, so one saturated chain and one unsaturated chain that kinks, and that allows some flexibility, but it's also separated by these other little molecules in between. Those little molecules are steroids. Steroids are composed of hydrogen and carbon, so they are a hydrocarbon ring structure, but they have this little tail right here. And they are important components of cell membranes because they protect the membrane fluidity, meaning they protect it from getting too stiff or too wobbly. So if it's really hot, you don't want your cell to melt, basically. So we have these steroids that are a heat protectant that prevent the phospholipids from breaking apart in the heat. But also if it gets too cold, we don't want our cells to become super stiff. So these kind of break apart the different sections of the cells, allowing for flexibility even in the cold. So these steroids are very important to membrane fluidity, but also cholesterol is a steroid that we find inside of our body. And we need cholesterol as a precursor to create vitamin D, testosterone, estrogen, progesterone, aldosterone, and cortisol. So all these different types of hormones are cholesterol based. So here's another picture of our phospholipid bilayer. If we have all unsaturated fatty acids, the bilayer becomes too fluid and breaks apart. If they're all saturated, they're too viscous or too stiff. So we have one saturated and one unsaturated, and they're broken apart by these little steroids in between. And the steroids will help to protect the cell membrane from being too hot or too cold or too fluid and too viscous. So cholesterol is really important here. Let's switch gears and talk about proteins. Now, I already mentioned that proteins are long chains of amino acids. So right now we just know amino acids are the building blocks of proteins. Amino acids are all made up of the same cell structure. It's going to be a central carbon with a hydrogen and an amine and a carboxyl group. Okay, an amine and a carboxyl group. But it's going to have something called an R group coming off of the side of it, which makes it unique. So you notice that each one of these amino acids in this picture look exactly the same. It has a central carbon, it has a carboxyl group right here, and an amine group right here. That's why we call them amino acids. This is carboxyl and this is amine. But then it has this chain coming off the end of it. And you notice that this chain is different than this chain is different than this chain. This is the R group. And this is what's going to determine the properties of the amino acid. And when we link a bunch of these together, it kind of changes the properties of all of the amino acids. Because, for instance, the cysteine is going to have this R group that likes other cysteines. So we might create like a fold or a bond here between these cysteines. It also helps with folding because some of them are maybe very electronegative and they bend away. Maybe they're very electropositive and they bend towards it. Maybe this R group is very hydrophobic and hates water, so it's going to fold inwards. Maybe this R group is very hydrophilic and it loves water, so it's going to fold outwards. And we'll talk more about protein folding later. But when we get a whole bunch of amino acids bonded together, we call it a polypeptide. That's because they're bound together with a peptide bond. Earlier, when we were talking about fats, we were talking about a glycosidic bond or an ester bond. Now we're talking about peptide bonds. And it's still through this dehydration synthesis. So we're still using dehydration synthesis. We're pulling the water out and creating this very strong, high energy peptide bond. Now, the amino acids come in a lot of different forms. There's 22 amino acids, and you've heard the word essential amino acid. That's because we can't create those 11 essential amino acids, or we can't create those, but we do create the rest of them. But they all have unique properties. Again, we have essential carbon with a hydrogen. We have the amine group, which is made up of a nitrogen, and we have the carboxyl group, which is the carbon connected to a hydroxyl. But the R chain will determine its properties. So the non-polar will not mix with water. They are hydrophobic, such as glycine, alanine, valine, cysteine. All of these, tryptophan, they are hydrophobic. They are non-polar. So if this is in your long chain, it's going to fold inward. These are polar, meaning that they're perfectly fine being close to water, and so these will normally fold to the outside. Serine, tyrosine, glutamine, these are going to fold to the outside. You might notice some of these are familiar. They do have different jobs within your body besides just building proteins, but amino acids' main function is to build proteins. We also have some positively charged amino acids and some negatively charged amino acids, such as glutamic acid or glutamic acid and aspartic acid are very electronegative. So they may create a very electronegative area on your protein, which would then attract a very electropositive, like lysine or arginine. So all of these amino acids have different properties, but they're still going to bind together through those dehydration bonds on the carboxylic to amine group. So this OH and this H will bind together, linking them in long polypeptide chains. Once they're linked together, they go through their secondary structure. And this is just based on the nature of those amino acids. Your protein is going to automatically fold up based on those structures. You can also easily denature a protein or break apart a protein, particularly with like heat or acid. Heat and acid will denature it. Just think of any way that you would cook your meat. You cook it with heat. You might cook it with an acid, like a ceviche. So there are different folding structures of proteins. The primary structure as a protein is built is the polypeptide. It's a long chain of amino acids with no folding. This is its primary structure, and this is called a polypeptide. From there, it will automatically fold in certain ways depending on the properties of those amino acids, either into an alpha helix or a beta sheet or pleat. So it could fold back and forth depending on those amino acids, or it could spiral depending on those amino acids. And this just happens naturally. Then we have a tertiary structure where the folding gets more complex. This may happen automatically, but usually we need another type of protein to help and fold this tertiary structure. Now, some proteins are functional at their tertiary structure, but some aren't functional until they become this quaternary structure where there's more than one peptide kind of linked together through its folding and through its properties. And they do usually need assistance to get to this level. When we talk about proteins being made later on in the semester, we'll talk about what structures help it to get to this quaternary structure. But this is a fully functioning globular protein, and a globular protein will have a specific job. It might be an enzyme. It might be an enzyme, it might be for transport, it might be a hormone, it might be for contraction, like actin and myosin are muscle fibers. It also might be for storage. It might be there to store protein or to store cholesterol. So your proteins have many different functions, and it will depend on their structure, how they're formed. The order that those polypeptides are bound in will determine its job. Okay, and here we're talking about what could cause a protein to denature. And this just means that we still have the polypeptide, but it doesn't have a functional form because it's kind of unfolded because we've broken or denatured it. So the folded protein will unfold or denature in the presence of acid or heat. So looking at this page, looking at this image, we can see lots of things that we've talked about here. We see carbohydrates. So this is a glycogen carbohydrate. Remember, we said that glycogen is a branched chain carbohydrate normally found in animal cells. So this represents glycogen. I can tell it's glycogen because of its branched structure, and also because this looks like a little glucose, a 6-carbon ring. Steroid. Here we can see these little yellow items right here are steroids. And I know that they're steroids because they're the shape. They have these 3 rings and then a little tail. And also it's embedded in between the phospholipids. We have several different types of proteins. We have globular proteins, which are shaped like this. They're globular. And some of them have the job of transport, so they look like a little tunnel right here. Some of them have their surface proteins. They're for identification, and they have this glycogen attached to them. Some of them are messenger proteins, where they will have some sort of receptor at the top, and then they will send a signal in the bottom. They have all kinds of different jobs. There's many, many different types of proteins, including structural proteins, like these little fibers. These fibers here are structural proteins. And then, of course, we have our phospholipids, with our hydrophilic heads and our hydrophobic tails. So we won't find any water here in the middle of the phospholipid bilayer, but the extracellular fluid is watery. It has water on the outside, extracellular, and the cytoplasm, the intracellular fluid, or inside the cell, is also watery. Let's talk about DNA real quick. DNA is made up of nucleic acids. So we have a long chain of nucleic acids linked together through a sugar phosphate backbone. So when you see this structure, this ribbon represents the sugar phosphate backbone, which just means the sugar and the phosphate kind of linked to each other. And then the active group, the nucleotide, is going to be facing in the middle. So nucleic acids are a class of organic compounds such as DNA and RNA, and they're called nucleic acid because they contain nucleotides, which are nitrogenous bases. See, nitrogen, 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 lots of nitrogen. And you should recognize that word adenine because that's the same group that's in ATP, except ATP has 3 phosphates, and this adenine structure, or adenosine, only has 1 phosphate. We have 4 different nucleic or nucleotides in DNA. We have adenine and thymine, guanine and cytosine. That is not supposed to be genuine. That is guanine. That's misspelled. Guanine, right here, guanine. I will fix that. If we're talking about DNA, if we're talking about RNA, RNA does not have thymine. Instead, it has something called uracil. So here's a more detailed structure. Here we can see adenine and thymine, and they both have these hydrogen groups facing outward, and they're going to bind together using hydrogen bonds, and that's what holds DNA together. We have 2 ribbons of DNA, and they're held together by these hydrogen bonds. That's why adenine always binds to thymine or uracil, because they create 2 hydrogen bonds. Whereas guanine always binds to cytosine because they have 3 hydrogen groups facing the middle, and they create 3 hydrogen bonds. These are very easy to break, so during replication they just unzip and then re-zip back up. Here we can see adenine bound with thymine, and here is a better image of that phospho-sugar backbone, the sugar phosphate backbone. Here we have, looks like the sugar connected to the phosphate, connected to another phosphate, connected to another sugar, connected to a phosphate, connected to another sugar. And this is DNA because we have deoxyribose sugar. So if I were to bind or finish this DNA, adenine only binds to thymine because of those 2 hydrogen bonds. Thymine only binds to adenine for those 2 hydrogen bonds. Guanine only binds to cytosine because it has 3 hydrogen bonds, and cytosine only binds to guanine because of the 3 hydrogen bonds. So it should look like this. Now, the difference between DNA and RNA are pretty striking. DNA is a double helix. It has 2 ribbons, and those nitrogenous bases are bound together by those hydrogen bonds. We have cytosine, guanine, adenine, and thymine. Well, RNA only has 1 phosphate sugar backbone, and usually the nucleic acids or the nitrogenous bases do not bind to anything because they need to be available for other purposes, like transcription and translation. And I want to look at thymine. If you look at thymine and you look at uracil, they look very, very similar. And so uracil takes the place of thymine inside of RNA instead of DNA. So that's one difference. The other difference is that the sugar in RNA is called ribonucleic acid, hence RNA, and the sugar in DNA is called deoxyribo sugar. So the sugar is the ribose, but this one is deoxyribose and this one is ribose. RNA comes in different forms. It's not just a copy of the DNA. RNA has many different jobs in the body. In fact, there's 11 types of RNA. But the main types that we're going to go over is messenger RNA, which is a copy of the DNA instructions that's allowed to leave the nucleus. DNA is not allowed to leave the nucleus. We also have ribosomal RNA. Ribosomes help to translate the RNA into a protein. So these are our protein building little molecules, little enzymes here, and they're made up of proteins and ribosomal RNA. And then we also have tRNA or transfer RNA. And transfer RNA are really important for protein synthesis because they carry amino acids on the end of them. So this is a little amino acid, and there will be a specific amino acid connected to a specific sequence of RNA at the bottom. So the difference between DNA and RNA, make yourself a T-chart in your notes. DNA is called deoxyribose. RNA is called ribose, or ribonucleic acid. DNA provides instructions for every protein in the body, but RNA carries out the orders of protein synthesis. DNA has a double helix. RNA has a single strand. DNA has GCTA, or guanine, cytosine, thymine, and adenine. And RNA has guanine, cytosine, uracil, and adenine. DNA can never leave the nucleus, whereas RNA is throughout the cell. RNA is everywhere. So DNA is kind of like having your blueprints filed at City Hall, whereas RNA is like the contractors come and make a copy of the blueprints to go construct things in the city. This is kind of an overview of the difference between DNA and RNA. DNA stays inside of the nucleus, and then we unzip it to make a copy of that RNA, and that RNA is allowed to leave, which then is used for protein synthesis. Here we have that transfer RNA, or that tRNA, bringing in amino acids to that ribose, or I'm sorry, to the ribosome. Don't forget to print your chapter laboratory write-up and bring it to lab, or complete it by scanning the QR code. If you don't come to lab, then you do still have to complete the laboratory write-up by scanning the QR code. Don't forget that you have a chapter lab quiz every Friday, but you also have a lecture quiz every Friday, and this is found in Blackboard. If you do great the first time, you're good. Move on. If you don't do too hot, you get a second chance to take it. Don't take it without studying. One of the great study resources that you have is the chapter homework, but you also have these items, the textbook, the flashcards, and games to play. So thank you for joining me this week for our lesson, and I will see you next week.